So when we talk about tau inclusions in Alzheimer's disease or in any other tauopathies, and we test for this, we measure this, we tend to use a specific marker of choice of the lab, usually related to phosphorylated tau. And this is our only measure of tau changes. However, several studies are showing us that the tau protein gets modified in different ways. So a phosphorylation in a certain site might be important, but it's not the only thing we see there. There are other pathological modifications. And this is called post-translational modifications because it happens after the translation. And nowadays, we have better antibodies to look into them. So again, thinking on how tau inclusions might kill the cell and be toxic to the cell, there are many theories out there. And one of them that came from animal studies that seems to have a lot of signal has to do with activation of caspase-6. Caspase-6 is a factor caspase, very often associated with apoptosis. And this activated caspase-6, already very upstream in the Alzheimer cascade, not downstream as the end stage of apoptosis, can truncate tau, can break tau in certain fragments. And these fragments, they are more prone to aggregation, so they become toxic. So what we wanted to test, or at least to map, is what is the situation with this caspase truncated tau in human brains? Is this important? Again, same idea. We see in animal models, doesn't mean that it's important in humans. We didn't have antibodies for this. So we partnered with Michelle Arkin here from UCSF, a very seasoned chemist, and we created monoclonal antibodies, and then we tested in the brain. And what we could show is that caspase-6 truncated tau seems to be very important in Alzheimer's disease and in other tauopathies that have 3-repeat tau, like Pick's disease, but not important in tauopathies that have only the 4-repeat tau component, which is PSP and CBD. But more than that, it seems that a very large number of neurons that accumulate this very toxic truncated tau, they don't necessarily show phosphorylated changes. It means that if we try to map Alzheimer's disease, just looking at phospho-tau or a specific time of phospho-tau, we are missing a lot. And this is not good because then we are not staging the disease properly. But more than this, we are not identifying properly all the neurons that are vulnerable. So if we believe that understanding what makes a neuron vulnerable to AD, so we can try to create strategies to protect these neurons, it's important to find all of them. And this is what this study is showing now.